Hello, everyone. Today we're diving into a crash course on the molecular biology of the gene.
We're taking a deep dive into, well, the fundamental blueprints of life itself,
Yeah.
drawing insights from some foundational research and even current threats.
It's such a core field, and to start, I think we have to recognize that this knowledge isn't just, academic.
Not at all. It's absolutely central to understanding global health challenges.
Absolutely. Let's start with the real-world power of genetics, maybe using the example of measles.
Oh, that's a perfect one. For much of the 20th century, measles was just catastrophic. I mean, it was responsible for millions of deaths every single year, especially among children.
And then we developed an incredibly effective vaccine.
The success was so profound that by, 2007, measles was considered nearly eradicated in the United States. Scientists really believed we had conquered it.
But the threat didn't vanish. It waited. In 2014, we saw the so-called Disneyland outbreak.
Right. Measles was reintroduced by an infected person, and it just spread like wildfire. That one event led to 147 infections across multiple states.
It was such a stark reminder.
It really was. Measles is caused by a highly contagious virus, and as vaccination rates drop, that constant threat posed by these microscopic instruction sets, these viral genes, it just immediately resurfaces.
And that sets the perfect stage for the big question, the one that drove all the pioneers of molecular biology.
What exactly is the chemical basis of heredity?
How do these instruction manuals pass down traits, whether it's from a parent to a child or from a virus to a host cell?
That whole quest, you could say, really began in 1928 with Frederick Griffith, a British medical officer.
And he was studying pneumonia bacteria, right? Two different strains.
Exactly. One strain was harmless and the other one was highly pathogenic, so disease-causing.
And he performed this experiment that, on the surface, just seems to defy all common sense. He killed the dangerous bacteria with heat.
Right, they're dead.
Then he mixed those dead cells with the living but harmless bacteria.
And the shocking result was that the harmless bacteria were, well, they were transformed. They suddenly became pathogenic.
Wait, so the dead cells somehow changed the living cells?
Not only changed them, but that change was permanent and heritable. They passed this new, dangerous trait onto all their future generations.
How did he even explain that?
Well, he didn't know the exact chemical. He just concluded that some stable chemical substance from the dead pathogenic cells survived the heat and was transferred, causing a heritable change. He named it transformation.
So it was the first real evidence that the blueprint was a chemical, but we just didn't know which one.
Exactly. And for decades, the debate was, is it protein or is it DNA? It took until 1952 for Alfred Hershey and Martha Chase to finally settle it.
Their experiment was just beautiful in its simplicity.
It was. They used the perfect model organism, a bacteriophage called T2.
Which is a virus that only attacks E. coli bacteria.
And it's so minimal structurally. It's basically a protein shell wrapped around some genetic material. The question was, what gets injected into the host cell? The shell or the stuff inside?
So their elegant solution was to use radioactive tracers.
Yep. They set up two batches. In batch 1, they used radioactive sulfur, which is only found in protein.
And in batch 2, radioactive phosphorus, which, importantly, is only found in DNA.
They let both sets of labeled viruses infect separate batches of bacteria, and then came the famous kitchen blender moment.
They agitated the mixtures to basically shear off the parts of the virus that were still stuck to the outside of the bacteria.
Like shaking off an empty coat.
Ah.
Then they used a centrifuge to separate things.
Yeah.
The heavy bacteria formed a pellet at the bottom, and the lighter viral bits stayed in the liquid on top.
And the results.
Completely definitive.
Right.
In batch 1, the radioactive protein, the sulfur, was found in the liquid. It stayed outside the cell.
But in batch 2,
The radioactive DNA, the phosphorus, was found inside the bacterial pellet. It had been injected.
the message was just inescapable. DNA is the chemical that enters the host and dictates the instructions. Protein is just the delivery system.
And that was it, the proof. DNA is the genetic material.
Okay, so with the material identified, the next massive scientific frontier was the structure. How is this stuff organized to store all that information?
Which brings us to the building blocks. DNA and its cousin, RNA, are polymers, which just means they're long chains of repeating units.
And those units are called nucleotides. Every single nucleotide has 3 parts: a phosphate group, a sugar, and then a nitrogenous base.
And those bases are the key. They come in 2 chemical families. The bigger double-ring purines, that's adenine or A, and guanine or G.
And the smaller single-ring pyrimidines, that would be thymine or T and cytosine C.
And before we get into the helix shape, We should probably clarify the two key differences between DNA and RNA.
Good point. They have distinct jobs.
Right. First, the sugar. DNA uses deoxyribose, which is really stable, perfect for long-term storage. RNA uses ribose, which has an extra little group on it that makes it more reactive, more temporary.
And second, the bases.
Yeah.
DNA uses thymine T, but RNA swaps that out for a very similar base called uracil or U.
Okay, so back to the big race in the early 1950s for the structure of DNA.
The double helix. And we have to start with Rosalind Franklin.
Absolutely. Her X-ray diffraction images, especially photo 51, provided the critical clues. They suggested DNA had a spiral or helical shape.
And crucially, that it had a uniform diameter all the way down. That uniform width was a massive hint.
It really was. It immediately implied there had to be two strands twisted together, a double helix.
So when Watson and Crick were building their physical model, that uniform width was their biggest constraint. If you try to pair two big purines together, say an A with a G, the molecule would bulge out.
And if you paired two small pyrimidines, like C with T, it would be too narrow. It wouldn't fit the X-ray data.
The only way to keep that perfect, uniform thickness was for a big purine to always pair with a small pyrimidine.
Exactly. And the specific pairings A with T and G with C were determined by the way they form hydrogen bonds. A and T form two hydrogen bonds.
While G and C form three, making that pair a little bit stronger.
And this elegant solution perfectly explained a mystery known as Chargaff's rules. For years, Erwin Chargaff had shown that in any organism's DNA, the amount of A always equals the amount of T.
And the amount of G always equals C. The Watson and Crick model showed why is because they're always paired together.
The best way to visualize it is as a twisted rope ladder. The sugar-phosphate backbones are the two long sides of the ladder.
And the base pairs, the AT and GC pairs, are the rungs of the ladder.
And one other key detail.
Yeah.
The two strands run in opposite directions. They're anti-parallel.
The beautiful thing here is that the structure didn't just show what DNA looked like. It immediately suggested how it could be copied so perfectly.
Which brings us to DNA replication and what's known as the semi-conservative model.
Semi-conservative. It sounds complex, but the idea is actually pretty simple. When the double helix replicates, each new daughter molecule is a hybrid.
It conserves one of the old parental strands and then synthesizes one brand new complementary strand. So half old, half new.
It's like using half of the original blueprint to guide the creation of a perfect new copy.
And this whole process is incredibly fast. It starts at multiple spots along the DNA called origins, opening up these little replication bubbles that expand and merge.
And it requires a whole team of specialized enzymes. The star player is DNA polymerase.
Right. That's the enzyme that actually adds the new nucleotides to the growing chain. But it also has another critical job. It's a proofreader.
A quality controller, so it checks its work.
It does. If it adds the wrong base, it can back up, cut it out, and replace it with the correct one. It's incredibly accurate because of this.
And since the process creates these short DNA segments on one of the strands, you need another enzyme, DNA ligase, to come in and act like a molecular glue.
It just connects all those pieces together into one continuous, seamless strand.
Okay, so we know what DNA is, what it looks like, and how it's copied. But what is its actual job? What's a gene?
Well, the concept of the gene has evolved quite a bit. It started way back in 1902, even before we knew DNA was the stuff of heredity.
With Archibald Garrod.
Right. He proposed that genes dictate our traits, our phenotypes, through the action of enzymes. He studied a hereditary disease and suggested it was caused by the inability to make a specific enzyme.
That idea was really cemented in the 1940s by Beadle and Tatum. They used bread mold, of all things, to develop the famous one gene, one enzyme hypothesis.
We've since tweaked that definition a bit. The modern view is that a gene is a region of DNA that is expressed to produce a final functional product.
And that product can be either a polypeptide, which folds into a protein,
or in some cases, the final product is just a functional RNA molecule itself.
This leads us right to the heart of it all, gene expression, the molecular chain of command. How do you get from the 4-letter language of DNA to the 20 amino acid language of proteins?
Well, you need a code. Since you only have 4 DNA bases, A, T, C, and G, and you need to specify 20 different amino acids, a 1-to-1 or 2-to-1 code wouldn't work.
But a triplet code would. 4 bases, taken 3 at a time, gives you 64 possible combinations. More than enough.
And we call that triplet code a codon. And what's truly amazing is that this genetic code is nearly universal across all life on Earth.
From the simplest bacterium to a person, that's a huge deal.
It's fundamental. and is what makes modern genetic engineering possible. We can take a human gene and put it in a bacterium, and the bacterium can read it perfectly.
So the first step in expressing a gene is transcription.
This is the process of making a temporary RNA copy, a message, from the permanent DNA template. The main enzyme here is RNA polymerase.
It binds to a start sequence on the DNA called a promoter.
Yep. Then it unwinds the DNA just a little bit and synthesizes a complementary RNA strand, swapping in a U, a uracil, wherever there would have been a T, a thymine. It just keeps going until it hits a stop sequence called a terminator.
Now, in more complex eukaryotic cells like ours, that initial RNA transcript isn't ready to go yet.
That's right. Before it leaves the safety of the nucleus, it has to be processed. First, protective elements are added. The special cap goes on the front, and the long tail of A's is added to the back.
These help it get exported from the nucleus, protect it from being chewed up by enzymes, and help the protein-making machinery find it.
And second is a process called RNA splicing. This is fascinating. Eukaryotic genes have these non-coding stretches called introns that interrupt the actual coding parts, which are called exons.
So it's like a film script with a bunch of random director's notes in the middle of the dialogue?
That's a great analogy. Splicing is the process of cutting out all those non-coding introns and stitching the coding exons together into one continuous readable message.
And what's really powerful is that a single gene can be spliced in different ways to produce multiple different proteins.
Exactly. It dramatically increases the complexity that our genome can code for.
Okay, so that mature messenger RNA, or mRNA, leaves the nucleus. Now it's time for translation.
Turning the nucleic acid sequence into a protein sequence. And for this, you need two key players, tRNA and ribosomes.
Transfer RNA, or tRNA, is the molecular interpreter, the bridge between the two languages.
It has to be. At one end, it carries a specific amino acid. At the other end, it has a 3-base sequence called the anticodon, which is complementary to the mRNA codon.
And the ribosomes are the factories where the proteins are actually built.
They're complex structures made of protein and ribosomal RNA, or rRNA. They have a large and a small subunit that clamp together on the mRNA. And the two main docking bays for the tRNAs, the P site and the A site.
The P site holds the tRNA with the growing polypeptide chain, and the A site is the entry point for the next tRNA.
The whole process kicks off with initiation. The small ribosomal subunit binds to the mRNA, and the first tRNA, which always carries the amino acid methionine, binds to the start codon, AUG, in the P site. Then the large subunit joins the party.
Then the real action starts, elongation. It's a 3-step cycle. First, the tRNA with the correct anticodon for the next mRNA codon lands in the A site.
Second, the whole growing polypeptide chain detaches from the tRNA in the P site and forms a new peptide bond with the amino acid that just arrived in the A site.
The chain just moved over.
Yep. And third, the ribosome moves. It translocates one codon down the mRNA. This shifts the tRNA that was in the A site over to the P site and kicks the empty tRNA out. The A site is now open for the next cycle.
And this just repeats over and over again, adding one amino acid at a time at incredible speed.
Until termination. When one of 3 stop codons UAA, UAG, or UGA enters the A site, no tRNA can bind. Instead, a release factor binds.
And that causes the whole complex to fall apart. The finished polypeptide is released, and the ribosome subunits dissociate, ready to find another mRNA.
It's an astonishingly precise mechanism.
But it's not foolproof. Tiny errors can happen, which brings us to mutations, any permanent change in the genetic information.
And a classic example is sickle cell disease. It's caused by a change in a single nucleotide pair in the DNA for hemoglobin, just one letter.
And that single DNA change results in a single amino acid change in the protein. But that's enough to completely alter the shape and function of red blood cells.
Mutations can be simple substitutions. Sometimes they're silent, meaning they change the DNA but not the amino acid. Sometimes they're missense, like in sickle cell, where they change one amino acid to another.
And then there's a nonsense mutation,
which is much worse. That's when a substitution accidentally creates a stop codon in the middle of a gene, resulting in a shortened, usually non-functional protein.
But perhaps the most disruptive mutations are frame shifts.
Oh, definitely. These are caused by inserting or deleting 1 or 2 nucleotides. If you think of the code as 3-letter words, this is like deleting 1 letter from the start of a sentence.
The entire reading frame shifts, and everything downstream is just gibberish.
These changes can happen spontaneously, just errors in replication, or they can be caused by mutagens, things like X-rays, UV light, or certain chemicals.
Speaking of things that mess with genetics, let's circle back to viruses. How do they replicate? Bacteriophages show us 2 main strategies.
The first is the fast and furious one. The lytic cycle. The virus injects its DNA, immediately hijacks the cell's machinery, makes hundreds of new viruses, and then causes the cell to lyse or burst open, releasing them all.
The kill and release strategy.
Exactly. But there's also a stealth strategy, the lysogenic cycle. In this case, the viral DNA inserts itself into the host chromosome. It becomes a prophage,
It's just a silent passenger.
a silent hitchhiker. It gets copied every single time the host cell divides, spreading the viral DNA without killing anything, until, that is, some kind of environmental stress triggers it to pop out and enter the destructive lytic cycle.
And those dormant prophage genes can be nasty. They can sometimes instruct the bacteria to produce dangerous toxins, like the ones that cause diphtheria.
And we keep seeing these emerging viruses like Ebola or Zika that seem to pop up out of nowhere. There are a few key reasons for this.
First is rapid mutation. This is a huge deal for RNA viruses, like the flu. RNA replication doesn't have that proofreading mechanism that DNA replication does, so they make a lot of mistakes.
Which is why the flu virus is a constantly moving target, and why you need a new vaccine every year.
Second is species contact. Viruses can jump from animal hosts to humans. H1N1, for example, circulated in pigs before it caused a human pandemic.
And third is just global spread. A person can travel from an isolated area to a major city in less than a day. Our modern world allows a local outbreak to become a global problem very, very quickly.
Finally, let's look at bacteria one last time. They reproduce asexually, but they have incredible genetic diversity. How?
They have three main ways of sharing genes horizontally. The first is transformation, which we saw with Griffith's experiment, just picking up naked DNA from the environment.
The second is transduction. That's when a virus accidentally transfers bacterial genes from one cell to another.
And the third is conjugation, which is kind of like bacterial mating. Two cells form a physical connection, and one transfers DNA to the other.
And this transfer often involves plasmids, these small circular DNA molecules that are separate from the main chromosome.
Right. And the really critical ones for us today are R plasmids. The R stands for resistance.
These plasmids carry genes for enzymes that can destroy antibiotics.
And because we've used antibiotics so widely, we've created this immense selective pressure. The bacteria carrying these R plasmids are the ones that survive and multiply.
Which is the molecular basis for the crisis of antibiotic resistance we're facing today.
So to sum it all up, we've gone through the entire molecular chain of command. The sequence of your DNA, your genotype, dictates an RNA sequence through transcription.
That RNA sequence then dictates the amino acid sequence of a protein through translation.
And it's those proteins that do the work, creating the cell's structure and function, which ultimately produces the physical trait, the phenotype.
It's just this incredible cascade of complexity, all built on 4 simple letters. But here's something to reflect on. We mentioned that the genetic code is nearly universal. The same dictionary is used to read the DNA of a bacterium and a human.
So if the same AUG codon means start for basically every living thing, and the same codons specify the same amino acids across kingdoms, what does that shared fundamental molecular language really imply about the common ancestry of all life on Earth?